Hi everyone, Jeffrey here, and today I'm going to be talking to you guys about the epithelium. Let's dive right in. So, when you're taking a look at the epithelium, it's broken into several major parts. And you can see that this is the apical domain, it's on top, and this is going to be the basal domain on the bottom, okay? And what you're looking at is the major parts of the epithelium, which is the Zonula Occludens, the Zonula Adherence, your Macula Adherence, and your gap junctions. So if we take a look at the electron micrograph right here, this is also going to be the apical side, and this is going to be the basal side of the cell, okay? And you can see here that these are going to be the Zonula Adherence, and this thing right here is just showing you the general area of where that's going to be. And then as you move further down, you can see that this is the Zonula Adherence. And then finally, you get down to the Macula Adherence. You can see that this is characterized by a little bit of a darker stain on the electron micrograph here. So that's the basic structure. Let's dive a little bit deeper. So let's just have a little bit of a review. Okay, let's do this together. I labeled these 1, 2, 3, 4, and 5. Just take a minute, pause the video, see if you can figure out what is what. Okay. All right. So now that you've paused the video and you've labeled it, number 1, this is going to be your microvilli. And the reason being is that you can see that this is going to be the lumen area because it's very light, and then you can see the microvilli sticking out right there. Number 2 is going to be the Zonula Occludens, right? So, and what you can see by that is it's characterized as being just the top part, the most apical part of the cell, the epithelium. Number 3 is going to be your Zonula Adherence, and that's characterized by a very dark spot on the electron micrograph. It's going to be posterior to the Zonula Occludens, and it's also characterized by having this clear spot right here. It doesn't have this line, okay? So that's how you can tell the difference between the Zonula Adherence and what number 4 is, and if you might have guessed, that would be the Macula Adherence. Now this looks essentially identical to number 3 except for this appearance of this line right here, which is also outlined by that arrow. Number 5, if you look at number 5, it looks very close to being number 4. Also, as it's more basal, you know that this is not going to be the Zonula Adherence at all. And it's also characterized by a darker spot right there. Okay. Now, let's move on into a more detailed look at each structure. So this is going to be what we said right here, your Zonula Occludens. You see the cartoon diagram right here. This is just a zoomed in version. The Zonula Occludens right here is going to be on the top of your epithelial tissue. And the reason for that is because it's formed of this occludin and claudin proteins, which form these types of junctions in the cell. And what these junctions do, and what the Zonula Occludens does, is that these junctions are called tight junctions. Okay, they're tight and they don't allow any water to pass through. They're very waterproof. They don't allow anything to pass through. So this allows for and it makes sense because it's on the top part of the cell, the epithelial cells, so that you're not letting water just freely pass through, right? And then you can see here, these little outlines are going to be your Zonula Occludens. Now, going down, this is going to be your Zonula Adherence, right? Because this is a little bit further down. It's characterized by that lack of um, that lack of a dark line in the middle. So as you can see here, the picture's not the best, but right here is going to be your zonula adherence, okay? So you can see here that it's made of ecadherin, and the molecules that make up these, the junctions are calcium, and these attach to actin filaments in the cytoskeleton. So that's a key difference here. Both of these attach to actin filaments, and then as we move further down, this, as you might guess, is going to be your macula adherence. Okay, and it's characterized by this presence of this dark line right here, which coincidentally is also called a macula. So that might help you remember it. And what's different between the macular adherence and the zonula adherence is that presence of that dark line, and also that it attaches to intermediate filaments in the cytoskeleton. So remember that these two both attach to actin, but this one attaches to intermediate. Now we move on to gap junctions. So this is going to be the cartoon of a gap junction. It's characterized by connexins. Connexins basically form this hexamer structure, and it allows for little pores to pass through. So you see this most commonly in things like neurons, where you allow for cell-to-cell communication, right? And this is an example. I also found another image that depicts this a little bit better. You can see that it pervades the membranes a little bit, the plasma membranes, and this is going to be your cell 1, and this is going to be your cell 2. So you can see that it allows for that cell-to-cell communication. All right, and the last thing I'm going to talk to you guys about is the basement membrane. The basement membrane, this is a very zoomed out electron micrograph. So if we take a look at this entire thing right here is going to fit right here, okay? So now we're looking at the basement membrane here, and it's essentially where your epithelial cells attach, and then they allow for the transfer of nutrients over. And this is a zoomed in photo of that. So this right here is going to be roughly this right here, and it's characterized by this thing of a basal lamina, and that anchors your basement membrane. Okay, so just to recap, the Zonula Occludens is attached to actin microfilaments. It forms a tight junction. That's the biggest thing for the Zonula Occludens. The Zonula Adherence, they also attach to actin microfilaments, and they form kind of a belt structure. So if you look back, this is kind of a belt because it's transmembrane, but it essentially functions as cell-to-cell adhesion, right? And then macular adherence essentially does the same thing, but with desmosomes, and it anchors itself to intermediate filaments. Okay. Gap junctions are characterized by connexins. They're important for cell-to-cell communication, and your basal lamina has laminins and type 4 collagen, and there's two layers. So it's the lamina lucida and the lamina densa. So if you look at here, the lamina, the basal lamina right here, the densa is actually going to be this dense portion right here. So that makes sense because it's dense and it's darker, okay? And then the lamina lucida is actually this white part right above the lamina densa. So hope that makes a little bit of sense. And then this is just another figure depicting those overall structures. You can see that these are different proteins that make up the complexes, and all the other functions that I've described a little bit before, okay? This is going to be your finger-like projections. So what that means is that you have these lateral indigitations, as they're called. Basically, this is going to be cell 1, and this is going to be cell 2. it's separated by this thing right here, and these are the lateral indigitations. And the reason why they go through this curvy structure is so that you have that increased surface area so that you can transport nutrients and things like that. Because remember, this is most common in your digestive tract and your duodenum of your small intestine, right? So you want to be able to transfer those nutrients, and much like the microvilli on top, you're trying to increase surface area so that you're able to get as many nutrients as possible. And then now I'm going to touch on briefly about focal adhesion. Essentially, this is just the cell, and it's trying to connect itself to the extracellular matrix right here. So, and it does that through this protein called integrin, and it's just, this allows for the cell to stay in one place. It doesn't allow it to move all over the place, and it allows for a little bit more of that organization. And then the last thing I'm going to talk about is hemidesmosomes. So what hemidesmosomes do, they're essentially kind of like your focal adhesion, but instead it's to your basement membrane. So this is going to be the basal part of a epithelial cell attaching to that basement membrane. So if we look here, this is going to be your epithelial cell, and then this is going to be your basement membrane, okay? And it's going to attach itself via these hemidesmosomes. And a hemidesmosome basically is just half of a desmosome. If you take a look at the desmosome structure, it has two sides right here, and the hemidesmosome only has one side, and it's very strongly just connected to the basement membrane, so it won't move. You know, you won't be able to kind of have an epithelial cell fall apart, because that would be really bad, since you're trying to get nutrients, but now you've lost the cell that has all the nutrients, right? I hope this video has been helpful. If you found it helpful, please leave a comment, leave a like, subscribe to the channel, and thank you. Have a good day.